Welcome back to the Learning Studio. This is the Biological Sciences series, and today we're starting a brand new section, Developmental and Plant Biology. Our first lecture in this section is on embryogenesis and developmental signaling, the biological journey from a single cell to a complex multicellular organism. If you missed our last episode on cardiovascular physiology, you'll find the full playlist linked in the description below. Here's a question. How does a single fertilized egg, barely visible to the human eye, develop into a body with a beating heart, a functioning brain, and trillions of specialized cells? The answer lies in the step-by-step miracle of embryogenesis and the molecular conversations known as developmental signaling. What is embryogenesis? Embryogenesis is the process by which a zygote, the fertilized egg, develops into a fully formed embryo. Key stages include fertilization. Fusion of sperm and egg creates the zygote. Cleavage, rapid cell divisions without growth, forming a ball of cells called the morula. Blastula stage, cells organize into a hollow sphere. Gastrulation, cells migrate to form 3 germ layers: ectoderm, mesoderm, endoderm. Organogenesis, organs and tissues begin to develop from these layers. Every animal, including humans, follows variations of this blueprint. Developmental signaling, cells talking to cells. For embryogenesis to work, cells must talk to each other through signaling pathways. Major signaling systems include ENT signaling, controls cell fate and body axis formation. Hedgehog signaling, guides organ and limb development. Notch signaling, regulates how cells decide their future role. BMP signaling, shapes tissues like bone and cartilage. Without these signals, development would be chaotic. Case studies and real-world sonic hedgehog, SH pathway. Named after a video game character, this pathway helps form digits in limbs. Disruptions can cause birth defects like extra or fused fingers. Thalidomide tragedy, 1950s to 60s. A drug once prescribed for morning sickness disrupted limb development in embryos, highlighting how delicate signaling is during embryogenesis. Stem cell research. Scientists use developmental signals to guide stem cells into becoming heart cells, neurons, or other specialized types, offering hope for regenerative medicine. Why this matters in developmental and plant biology. Embryogenesis explains how complex organisms are built from scratch. Understanding these processes is crucial for medicine, preventing and treating birth defects, infertility, and developmental disorders. Biotechnology, growing tissues and organoids in labs. Evolutionary biology, comparing developmental stages across species, shows how life diversified, past, present, and future. Past, early embryologists like Aristotle studied chick embryos, laying foundations of developmental biology. Present, advanced imaging lets us watch embryos develop in real time. Future, synthetic biology may one day allow us to engineer development itself, designing tissues or even artificial embryos for research. So that's our exploration of embryogenesis and developmental signaling, the journey from one cell to many, guided by nature's molecular instructions. In our next lecture, we'll shift from animals to plants, exploring one of the most essential processes on Earth, photosynthesis, light and dark reactions. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the next lecture. Because once you understand embryogenesis, you realize life itself is a carefully choreographed dance of signals and growth.